Welcome to the Phase Space Invaders podcast. Today you're listening to episode 27, where my guest is Kresten Lindorff-Larsen, a professor at the Department of Biology at the Linderstrom-Lang Center for Protein Science, University of Copenhagen. Many of you will know Kresten from his work on force field development, in particular the very popular Amber 99 ILDN, but his work extends deep and wide from integrating NMR data into protein ensembles, to studying the dynamic folding and unfolding of protein domains, to modeling intrinsically disordered proteins or protein regions. In particular, Kresten's work in the latter direction has been recently thrust into the spotlight with the recent coarse-grained model, Calvados, that brings us one step closer to capturing the properties of unstructured regions. But again, that's far from a complete picture of what he'd been working on. So we start by revisiting Kresten's path from a wet lab biochemist to a computational biophysicist, a story full of open-ended explorations and helpful mentors. Kresten gives us some background on how both the legacy and latest models developed, highlighting how in each case the driving force were experimental results that either weren't quite matching simulations or were plenty enough to allow for top-down training. We walk through some of the functions and applications of intrinsically disordered regions, or IDRs in short, and the relevance for medical research. Then towards the end, Kresten shares some tips and observations from his work in grant evaluation, insisting that internal peer review remains the best source of feedback. But in the end, it's one's scientific intuition that has to guide us. I hope you like our conversation today. So, Kresten Lindorff-Larsen, welcome to the podcast.
Thank you very much, and thanks for having me.
So, Kresten, you very recently made a bit of a splash with the exciting research that came out on intrinsically disordered regions with your latest Calvados model, but you're one of those people who really leave their mark on our field of computational biophysics in so many places. And, all that starting as a biochemist, not a physicist, although you've been working very close to physics in many of those projects. So going back to your days at the lab of Michele Vendruscolo, by the way, a former podcast guest, did you find this transition natural or daunting or exciting?
Great. I think all of the above. I was thinking, through this earlier today, and I really think that lots of the things that we do have happened because of chances. So I studied, as you say, biochemistry, and I did undergraduate work doing wet lab experiments, purifying proteins either from yeast or bacteria or from barley grains. And then I went to Cambridge to do my PhD with Chris Dobson. And Chris had just moved from Oxford to Cambridge, and so the lab was pretty non-existing at the time. And so I started doing wet lab experiments there in the beginning, but it was also clear to me that I wasn't really making so much progress because the lab was pretty empty. And so I started talking to Chris and other people at the department and ended up, talking quite a bit to Michele, and then did lots of work together with Michele and Chris during my PhD. So this was really a bit of a random point. And then I just had to sort of learn the biophysics and the simulation tools along the way. So very much by chance. And then I've sort of have been trying to catch up ever since.
Yeah, failed experiments account for a lot of great theoretical computational biophysicists in the end.
Yeah.
Do you have any tips for people who are doing this transition? Like how did you manage that? Was there a structured strategy for this conversion, or was it just haphazard?
I was very lucky to have great people supporting me. So Chris and Michele, Emanuele Pacci, all helped me out enormously in the beginning. And then I had already during my undergraduate studies, studied quite a bit of physical chemistry and statistical mechanics and some quantum chemistry also. So I was at least used to some of the more technical things and had a reasonable background in some of the theory. And so that made some of the things easier. But then I think the other thing that I did, and this was not really my own decision, this was again a little bit random, was that I ended up focusing on things that sort of bridged between the things that I knew and the directions I wanted to head. So since I had a background in biochemistry, it felt very natural to me sort of to try to work at the interface between simulations and experiments. And this, of course, was again something that Kresten, Michele, and Emanuele and others had pioneered a few years before. And so I ended up sort of in the right place at the right time, and not necessarily with the right background, but trying to build the knowledge that I needed to do these things.
Right. It feels like the interfaces are where most of the action is happening. That's definitely true.
And the other thing that I think was, again, helped this was sort of a very practical thing, which is I had a scholarship from the Danish government to go to Cambridge to do my PhD. And so I had quite a bit of freedom in what I wanted to do, which was great, including that I decided to spend my summers in Copenhagen. And so I arranged to have a desk together with one of my former supervisors, Professor Flemming Poulsen. And so I sat and worked from their lab over the summer. And so that lab was doing NMR experiments on, amongst other things, unfolded proteins. And so I ended up talking to Kåre Teilum and Birte Kravlund and others around the time who were then doing NMR experiments on things that I thought exciting. And so because I could spend some time in Copenhagen, I ended up picking up these experiments and experimental data and bringing the data to these joint projects with Cambridge. So again, this was completely by chance, but also maybe having the background to see that there were some interesting possibilities in doing this kind of collaborative work.
Right, I see. And that was also the beginning of the golden era of protein folding, right? So you ended up at the E-show soon after that, working on the actual folding of those small protein domains or like fast folders, and also contributed to the ILDN amber force field, which is still a cornerstone, like probably still my favorite force field to use for protein simulations. So how did that unfold at the time?
Yeah, that's a good point. So I was just looking it up, and it's exactly 15 years ago since it was published. Yeah, again, this is a little bit random. So if we take a little bit of a step back, I worked on protein folding quite a bit during my PhD. I was fortunate enough to be at a department, with Chris Dobson, Alan First, Jane Clark, and many other protein folding experts. And so this was always an interest of mine. And then I sort of got sidetracked into first studying the unfolded state of proteins, sort of trying to start the starting point as well as the transition state. And this was then work that I took with me when I moved back to Copenhagen after my PhD. So we worked quite a bit on studying both unfolded proteins and then also intrinsically disordered proteins that we can maybe return to. And as part of this, we developed a method quite early on learning force field parameters from experimental data. So we published a paper, one of the first papers when I started my own group after my PhD, on using sort of a Bayesian approaches to learn force field parameters against experimental data in sort of a top-down approach. And so then when I moved to New York, that was one of the things I was interested in. And I started a program where we tried sort of to set up these approaches. And in the end, some of those things worked pretty well. But again, a little bit by chance, we ended up being sidetracked into looking at these side chains and found that there were some of the side chains that looked to be not quite as good as we had hoped. And while I would have loved to have done this in sort of a top-down approach using, my background in experiments, we actually ended up parameterizing it by quantum methods. So we have, good colleagues that were experts in quantum chemistry, and then using a broad set of experimental data as validation. And so this was all, again, because we knew that working with force fields would be important if we're going to run these long simulations. We started to see some problems with deviations between simulations and experiments, and then use that as the starting point for tackling these force field problems. So again, and I think this is probably true for most people who work on force fields, this comes out not of an intrinsic desire to work on force fields, but because one discovers some problems, with the force field the body's using, and then you have to fix them in one way or another to do the research you'd like to do.
Yeah, it's a constant back and forth between applications and methods, right? The best methods come out of applications, and the best applications are what is at the forefront of the current experimental methods
I agree
capacities.
completely.
So yeah, that's a common theme across your career, which is, I think a great template for people who want to have their own fulfilling scientific careers. And so now we ended up with a working force field for IDRs, right? Like, you have this Calvados model, which is already in version 3. I remember we had a conversation about naming conventions and how you want to version the iterations of the force field. So let's talk about Calvados V3.
Yeah, it's
what can it do? Like what can it do that we couldn't do before?
Yeah, I don't know, to be honest. It's again a project that sort of started around 2005 or 6 with this work that I mentioned before. So one of the wonders of being at a university with lots of teaching is that you get to work with great undergraduate students. And so I had a master's student, Anders, who worked together with me, and then a colleague at the Niels Bohr Institute of Physics. And we then got to start doing this early development of learning force field parameters from experimental data. And back then, there was just not enough data really to do this with the breadth that we liked. So we did some work, we developed a coarse-grained force field, but there really was very little of the kinds of biophysical data that we would need. And then I sort of got sidetracked or worked on folding and other things. And then some years ago, again, I thought, maybe it would be time to pick up this project again. Ramon Crevet from Barcelona came on a sabbatical in Copenhagen. There was a great, again, undergraduate student here, Klaus Schulze, who worked with Ramon, and we picked this up. And then Giulio Tesei came into the group and joined the effort, really just trying to make a basic model that we thought could be accurate. And I think sort of the driving idea was, and that's been true for many things, is that experiments are in some sense infinitely accurate. Of course, there can be uncertainties in the measurements, but at least they report on nature. And often because, of Boltzmann factors, you can see deficiencies or small deficiencies in force fields. And so targeting experimental measurements can really fine-tune things quite a bit. So we had the idea we could do a relatively accurate coarse-grained model on a restricted set of problems, which initially was just to capture long-range interactions in disordered proteins. So that was sort of what now we call Calvados 1, although we didn't actually call it Calvados at the time. And then we had sort of a fine-tuned version that we then ended up calling Calvados 2 because, rather than do more fine-grained naming, as you mentioned. And I think this model is still great for studying disordered proteins. But of course, disordered proteins is an extremely broad term, right? It covers many, many different things. And disordered proteins is not just one thing or one biological function. And of course, they generally exert their functions together with folded domains and other macromolecules, RNA. And so it was pretty clear to us that if we wanted to have a model that we could use more broadly, we needed some way of bringing in the folded domains. And so the Calvados 3 work came out of that desire. We initially thought it would be easy. Then our initial ideas turned out not to work. But again, Sankar and Giulio and Søren von Bülow worked on this and figured out sort of a relatively simple hack that allowed us to bring in folded domains, at least in some primitive way. So these models are not perfect. There is a restricted set of things that they can do. But for those things, like characterize global structural properties, they work surprisingly well for a model that has one beat per residue, 20 parameters plus a simple model of charge and electrostatic interactions. And so, yeah, we're having a lot of fun with this model. It's really enabling us to do many things that we couldn't do before.
I see. I can imagine one of my experiences is that coarse-grained models work so much better with all the force field development tools that are supposed to be exact compared to all-atom models, right? Because with all-atom models, you have so many issues with sampling, with sensitivity and noise. And coarse-grained model, I believe, are just much better behaved in all these regards. So yeah, they're probably the best things to parameterize if you have a bunch of experimental data. compared to how hard it would be with an atomistic?
No, I mean, so we've also done that, right? So some of the work that in particular Stefano Piana and I were doing at the Shaw Research and later picked up by Paul Robustelli and others was, of course, targeting parameters in all atom force fields against experimental data, either directly or indirectly, and with either a little bit trial and error or with systematic approaches. But you're absolutely right. It's harder, not just because of convergence, but of course, because when there are more parameters, you need to make more decisions about where to fit. And of course, this is one of the problems of top-down parameterization, is that you may be, fixing the errors with the wrong terms. And of course, for a coarse-grained model where there's just fewer terms, there is in some sense less risk of messing up because there are not so many terms to fix. So these are, per definition, of course, effective parameters that capture many different physical effects. And as long as you know that and you try to restrict the problem set and have enough data, I think that, as you say, these things are at least more robust for this kind of top-down approach. And of course, now lots of people have combined bottom-up and top-down approaches via something the Martini framework, but also, of course, now with the automated all-atom force field parameterization that several groups are working on and doing tremendous progress on.
True. I talked to Paul, before in the podcast and recently here as well. And he's saying that, they have a few things that only atomistic force fields can do in IDPs, but he also recognizes how much sampling is needed for any kind of meaningful outcome to come out of that. And because of this coarse-grained approach, you could move on to what you called proteome level research recently, right? So like trying to look at the properties of the entire human proteome in a single study, which is impressive. Can you tell us what comes out of such studies? What kind of statistical trends or properties you can extract?
Yeah, so we were very happy with this Calvados model in the sense that it's relatively accurate for certain things. And as you say, it's also pretty fast. And so we started thinking about how can we use this speed to do new kinds of things. And we sort of took it in two different approaches. One is a project of IDP design that involved running many simulations, but one after another. And the other one is the project you mentioned, where we studied all IDRs from the human proteome and ran simulations of some 30,000 sequences. And so I think this was certainly a fun project. It allowed us to bring many different expertises in the group together because it allowed us sort of to bridge the molecular biophysics with more bioinformatics and even disease biology, things that we've also been working on. So in some sense, it's sort of enabling us to do molecular biophysics, but at proteome scales, and bridge it in with sequence-based machine learning, studies of disease variants, studies of functional effects of IDRs, and really asking things at a different scale than what we've been doing previously in our research. And this was a fun project, and we are doing more of these kinds. But I also think that this is something I see many other people in the field moving in that direction, still trying to work on individual systems and studying them at depth, but also trying to scale up to look at whole classes of proteins. Right? We just saw in a wonderful paper on simulations of a big chunk of G protein coupled receptors, studies of many kinases, or in our case, studies of all disordered things from human proteome. And then, of course, you can then go back and look at this data set as a whole and try to ask questions about the trends, across this, and what is the relationship between sequence and conformational properties, for example. And then we and others have used these things sort of to train simple or for, in other cases, less simple machine learning models to predict these properties directly from sequence.
All right, that sounds exciting. And also opening the way for new biology, right? I studied DNA binding domains, and there was always this feeling that you have a small domain that does something, it binds or doesn't bind. But then you look at the whole sequence, and it's like it's a monster that has a 50 amino acid domain and then 300 amino acids in this IDR, and that's connected to something else that connects yet to something else, right? And like I always had this question: okay, I'm studying some tiny fraction of this whole process or the whole structure that is being assembled there. But then, how does it really translate into the biology, right? Are we starting to uncover this, or to, like, for example, in the field of, transcription factors or RNA binding protein?
Yeah, I think a lot of people are making great progress on this. We're doing some work in this area, and lots of other people are trying then to reach these things from the molecular level to the interactions, to studying things that are, DNA binding at the chromatin level, and linking it to some more general effects on gene regulations. So yeah, there's this whole hierarchy of interactions and regulation mechanisms that I think we're trying to, as a field, to uncover. But I will also say one of the wonderful things of working at a department of biology that's extremely broad is that I get to see, on a first-hand basis, that there's always another scale up front, right? So we are sometimes, asked, why are you studying these things, in a computer, right? Biology is much more complicated. You should do cell experiments. And then if you do cell experiments, you're asked to do it, in an organoid or in a mouth or something. But of course, there's always a scale above that. Then there is, again, there's ecosystems and interactions between ecosystems. And so I don't think that there is a single scale that, covers everything, right? There's complexity, in many layers up and in many layers down. And as long as you realize that and have some understanding of where your research fits in, I think that, studies of individual proteins are still extremely important. And then you just either need to know enough or collaborate with the right people to put that in the context for, say, as you say, understand how do these proteins interact with co-regulators, or how do the transcription factors bind the DNA and bring in other things or open up chromatin. I think that all of these things are necessary. And I also see that lots of people are now trying to, really bring these things together in sort of a more holistic description. I think that's just wonderful.
Yeah, it definitely goes all the way. Do you have any interesting examples of function of IDR that goes beyond just, connecting to domains? Did you discover or uncover something that's really, really interesting in terms of, why this IDR has to be what it is to perform its biological function exactly, rather than just keeping things in proximity?
It's a good question. And my initial answer would be no, not because there's not individual IDRs that we like, but I think one of the things we also like is really to look at things across scales. So we do work on individual proteins, often in collaborations with people that, then do experiments. So I'm working with colleagues here at the department on RNA binding proteins that are involved in epigenetic regulations or in transcription factors. And then I'm in the wonderful position of getting to see glimpses of all of this interesting biology. But for myself, I'm really focused on these things as a general class of problems. And then I also think that and maybe this is to some extent the IDR field's fault, but, IDRs very generally do things together with the folded domains, right? It's not like one can always see them in isolation, right? So in our paper, we looked at them very concretely in isolation. And of course, this is a major limitation of that work. But yeah, they do things like, as you say, they bring in folded domains together, right? We worked on multi-domain enzymes where the relative orientation between binding domains and catalytic domains determine activity, or as we mentioned, transcription factors. Clearly, the DNA binding domains play roles in binding to the DNA, and then the activation domains help bind other things. And so I think one has to sort of look at them as a whole. But of course, many of these things don't work without the disorder or the regions, right? But they are complementary and do different types of interactions than their folded domains.
Right. So when you look at, say, mutations in IDRs, what kind of property changes would you be looking into?
Yeah, so this is something we've looked at quite extensively, and maybe not so surprisingly, you think of folded domains versus disordered domains. We and other people have found that missense variants that cause disease are generally, of course, enriched in the folded domains. They're much more fragile than the disordered regions. You can have many individual amino acid changes in disordered regions that have very small functional effects. And then if you look at the disordered regions, we and others have found that they are sort of enriched in the regions that probably then make specific interactions with other proteins in short linear motifs. You can sometimes discover these with AlphaFold. AlphaFold sees them as being more conserved. So there's about 15% of the disordered regions that may be fold upon binding, and disease variants are enriched in those. And then outside of those, there are lots of mutations in total number, thousands of them. But I don't think we as a whole, as a community, really understand whether they're general mechanisms or whether there are just lots of individual cases. So mutations that affect post-translation modifications. There's the mutations in the short linear motifs that I just talked about, but if you take those away, there may be mutations that increase aggregation. There is, of course, known in a bunch of diseases or missense variants that caused perturbation, for example, to the formation of biomolecular condensates. But again, they tend to be relatively robust. And so, for example, in one of the many supplementary figures in our paper, we sort of looked at the changes of the biophysical properties of missense variants that cause disease versus those that don't. And there was a statistically significant difference, but not really a difference that explains much of disease variants. And so I think we really need to have a much better understanding on what are the interactions that these things make, what are the relevant polymer properties that are important, and then use that to tease away biology. But it could also just be that there are few general mechanisms. So I think our work on folded proteins have shown that there are very general mechanisms, right? About 50% of missense variant folded proteins, they just mess up the protein structure and cause degradation. And maybe there are just not so many simple general mechanisms in disordered regions, but I think time will tell.
Right. Seems like the next, maybe not so low, but hanging fruit.
Yeah,
to grab.
Yeah, absolutely. No, I think that there's lots of people working on this, and of course, we're now starting to accumulate, more and more data and corresponding more and more simulation and other computational methods that can start to address these things. So both predict which variants cause disease, but also trying to peel away the molecular mechanisms of how they cause disease. And I think again, these kind of mods like Calvados and others, I think will hopefully play a useful role. I think in the IDR field, we're still a little bit hampered by the fact that we don't have the same amount of systematic data and tools that work so well for folded proteins. And so we need these kind of physics-restrained models to interpret the experimental data. This can either be simulation models or theoretical models, or combination of these with machine learning models that then allow us to look at these things at scale, but learning from limited or imperfect data in a way that maybe one can get away with not doing if you're looking at the folded domains. But yeah, I'm very hopeful, and I think it's also fun and good that many people are working on this.
Right. So back to incremental improvements based on whatever is available. That's a common theme. Yeah, also quite notably, you can now design IDRs, right? So you can use this model also to generate IDRs with specific properties. Why would you want to do that? Like what is the selling point for bespoke IDRs, for example, or IDRs?
Yeah, I think that there are a couple of reasons for doing that. One of them was, just the fundamental curiosity question, can we do it? And then building on that, saying, well, if a third of the amino acids in the human proteome are disordered, and there's lots of evolutionary and genetic and biochemical evidence that IDRs are used extensively by biology, they must be able to confer functions that are maybe not so easily performed with folded domains. And so say, if biology uses it, wouldn't it be great if we could also design these things sort of at will? And of course, people have designed IDRs before using sort of rule-based design methods, and they work very well. And what we sort of tried to do was say, well, maybe we can take sort of a more automated computational method where we sort of let the simulation tool design the sequences either directly via these brute force simulations, as I mentioned, or, using machine learning models as intermediate models. And I think it's an open question on, what exactly we'll do with them. I mean, we've not really done anything practical with these things yet beyond test our understanding of sequence ensemble function relationships. But again, if you want to design enzymes that target specific surfaces with, many of them, they have, long disordered regions that tether together substrate binding domains and catalytic domains. You have lots of receptors that have disordered regions. Maybe if you want to design orthogonal, signaling molecules, having some design protocol would be interesting. So I think it's really, an open field that is starting to take off now with multiple groups picking this back up again, both for sort of curiosity-driven research and as tool in studying biology, and maybe for sort of technological, applications of using them to improve enzymes or formulation or other things. But I think, in this case, we sort of started the project to see both, can we do it? And secondly, we thought, well, if we can design proteins that do not exist in nature, and then we can go make them, we can then compare them to the model, the simulation model, and then use that as a relatively stringent test on whether the model extrapolates well to a completely different sequence system from what we built it on. And I think this was nice to see that, yes, indeed, it worked very well also for things that look nothing like the sequences we have in nature.
Right, that's a great point. Can we expect to see attempts at IDP therapeutics in 5 years from now?
I mean, to some extent, the success of peptide hormones as drugs is an example of this, right? Many of these are very flexible.
Right.
I come from a country that produces lots of GLP-1 receptor agonists. And so these are certainly disordered molecules. And of course, lots of peptide hormones are like that. I don't know whether we will see IDP drugs in general. You might see drugs targeting disordered regions or interfaces between disordered regions and folded domains. But I don't think we are at a situation that where we necessarily want to develop IDP drugs. But of course, someone will then tell me that I'm wrong and go do this, and that would, of course, be great. No, I think that, we now can design folded proteins that have basically whatever structure we like, and there's much progress in also making them bind to other things and maybe even work as catalysts. And so that's, of course, solving a big part of what proteins do in biology. But then, of course, proteins are dynamic, and so people are working on putting in dynamics into folded domains so you can switch them on and off. And I think disordered proteins is just one of the tools, or disordered regions is one of the tools that biology uses to regulate other things. And so if we can, design those at will with whatever properties we want, then we can potentially expand the scope of protein design to all the same kinds of things that the disordered proteins do in cells, whether that be as enzymes, as I mentioned, or in synthetic biology as, transcription factors or what have you.
Right. It definitely goes both ways, as you mentioned, that people are working on designing drugs that bind to IDPs or IDRs as well. And I don't know how promising a field that is, but it is a field now.
Of course, I have a conflict there because I work with people that do this, but I will say that I think there's so many important proteins with disordered regions that if we could do it, this would open up lots and lots of possibilities. But as you say, and as many other people have said before, it is, of course, an incredibly difficult endeavor. We haven't really explored it that much and very systematically yet, so I think it's still pretty open how far this can be pushed and what the tools and the ideas are. And I think it's great that someone is doing this. I think these are the kind of problems that we will only really find out by trying. I think it's very difficult to predict whether this will be successful or not. In general, and of course, in individual cases, it can be very successful.
Right. It feels like, as you say, it's hard for our community to tackle it. Maybe it's easier to do in those broad screenings that are done experimentally. I mean, you have a lot of biochemists and experimentalists around you, right? So maybe it's a question to your collaborators rather than to computational biophysicists.
Yeah, or I think as with so many other things, as sort of finding the right interface between computational methods, simulation methods, machine learning methods, and the right kind of experiment that have the accuracy you need and the scalability that you need. And again, you can do lots of things with phenotypic screens, but then it's very difficult to figure out exactly what you're hitting and the mechanisms. So I think all of these things, they really have to come together to address these questions. And that's, about having the jobs that we have.
Right. I can clearly see the role of computational biophysics in, say, lead optimization, but not maybe lead design or like coming up with the initial scaffold for something that binds to something unstructured, right? But you can probably guess what you can add to a molecule to make it stick more to some kind of stable mode of interaction.
Yeah, I don't know, but I'm very happy that someone is trying to tackle this and build the data and the understanding and the tools to do this. And clearly our co-screen model working at the resolution of 1 bead per residue doesn't have in itself the chemical accuracy that's needed to study the effects of individual hydrogen bonds, but maybe together with other tools, these things can play a role. And then I think, of course, that the work that we and many other people are doing are trying to look at biological problems at a larger scale. Hopefully can also help understand some general rules of where to target things, or what are the kinds of sequence motifs, specificities one might necessarily, one could potentially address.
Yeah, right tool for the right question. You alluded to that before, that when you do grant evaluations, very often this is a huge consideration that people often skip, right? Like they try to approach a big question and then they end up tackling completely unrelated subfield or sub-area of that question. What are your guidelines or suggestions or tips for people who struggle with or generally write grant applications to make them both having a higher chance of success and maybe making it better at addressing actual questions?
Yeah, it's a difficult problem and question. And I think lots of people will tell you, including me, that you should try to put yourself in the shoes of the person who will read it, which of course already brings up the first problem, which is that you don't know who will read it. And secondly, we're all individuals with our own pet peeves and niches and expertises, right? And so there is some randomness in this process simply because the people who will read the grants, even if it's multiple people, will be individuals with expertises and explicitly or implicitly things we like and dislike. But that said, I think it's important to try to write things so that you explain why what you're doing is important. I think that's sort of, in some sense, obvious. But then I think what I sometimes see lacking is then the connection between the actual proposed research and the bigger research questions that you set up. Right? So in some sense, you write your introduction and your overall research area and aims, and then there will be some middle part that explains how you will do this. And then as an evaluator, I would like to see that if everything is successful, at least then that should provide some insights or progress towards the big prep question that you set up in the beginning. And sometimes I find that this can be missing, that people, they talk about some big overall problem in biology or physics or whatever the topic is, and then they go right about what they're going to do. And then they don't really help, make the connection to how this will move the field forward. And of course, sometimes I think it's because people don't think it through. I think more commonly, it's just that they think this is obvious, but maybe it isn't obvious to a reviewer who has a slightly different background or who doesn't know the field. And so I think this is one of the more common things that I would encourage people to think a little bit more about, sort of, really connect the whole arch from the idea to, to what you'll actually do to where this will move the field forward. But then in general, make it an easy and reasonable thing for the reviewers to read. But I will say that it's a lot of time it takes, but I really enjoy reading grant reviews. I learn a lot, both about science and how people think. And also, I think, learning how people are trying to connect ideas. And it's also good to see that there are many ways of writing good grants. There's just not, there's not a single way that this, must be done. And I think this is always useful to sort of see different ways of how things can be done, because of course I tend to, I think, my own way, and that's not necessarily the right way. So that's sort of some high level views. And then, even if one is successful in the sense that you're running with the average success rate, it still means that most grants that you will submit will be rejected. And so the other recommendation I have, unfortunately, is just to submit many grants, which can feel frustrating. But with the way that things operate, that's probably the way that we have to go for a while.
Yeah, this is how you win the lottery. You buy a lot of tickets. It's a fair point. But also, I think, you have this cross-disciplinary experience, and I think a lot of people in the field lack it, perhaps me included. I mean, it's pretty intimidating to go from a computational model to like an actual biological outcome, right? For most of people who are on the theoretical side. So there's also a question of how we build this kind of marketplace of ideas where people can give each other feedback and like improve or add to. Should grants, for example, should they have rounds where you would improve your proposals with each feedback? Because sometimes it feels like people just submit the same application and it's either successful or it just goes down the drain, right? Like how much you could maybe improve if grant applications were more like papers that you submit and you revise and maybe something interesting comes out of the feedback.
Yeah, I think that certainly you can get feedback that can improve things. Of course, these kind of things exist to some extent in different funding bodies, in the sense that you do get feedback, and sometimes, it's even useful, maybe not always so useful. I think then it runs into the problem that, of course, next time you submit, it might be a different group of people that review it, or you might be competing with a different set of alternative proposals. And so something, if you're told, well, this is great, but if you cut away this part and that this other part, it would be even better. Then if you do that, you might end up with a different reviewer or set of reviewers that say, well, I don't really like this, or they will say, this is great, but there were others that were even better. And so I'm not sure that there is a simple way of doing that. So I think at least this is what I recommend, is that, yes, it's great to get feedback, but the best way of getting feedback on grants is to show your grant proposal to your colleagues, and maybe not just the colleagues next door, although that can be great, but maybe also someone who is a little bit further away from your scientific discipline. Now, in my case, the colleagues next door are sort of, a little bit further away, but I think it's both good to get some detailed feedback, but also from someone, who's a little bit further away, because, in most cases, the reviewers will often be people who are not experts on the things that you like. And so I think it's always great to get feedback on your proposals, and ideally from as many people as possible. We established something here at our department where we were a group of people that offered to provide feedback to grants. Of course, people end up doing things last minute, which of course makes this hard sometimes, me included. But I think this is something I can very recommend, to get feedback and to listen to it, but also at the end, trust your own instincts, because of course you are also yourself the expert on the projects.
All right, so peer review stands the test of time.
I mean, it is a good mechanism, and to be honest, sometimes it just makes you rethink things. I just say, you thought you wrote something perfectly clear, and then someone who should know this reads, and then they don't understand what you're writing, and it's clearly because you made assumptions on behalf of the reader that were not correct. And I think it's just useful to get that up front. I will say, that you mentioned these things of interdisciplinary research projects, and it is hard, and I've seen it up front, both when I've sat on the ERC panel and also in my current panel, is that these projects can sometimes have a hard time. I mean, they can also fly through because many people are excited about it. But I think, if you don't really fit the target audience, many people that may think that this sounds interesting, but maybe are not really enthused about the specific details of that project. We had one project that, was funded finally, but that was evaluated as an ESC project, both in the physical sciences and the biological sciences. And I think that, they got really nice reviews and very thorough. I have no complaints about the reviews. They were really good. But I also got the impression that the physicist maybe thought it was very biological and the biologist thought it was very physicsy and weren't as enthused about it as I were. And maybe it wasn't written, as well. But I've seen this both, as an applicant, but also as an evaluator, that sometimes these things can have a hard time.
Yeah, I can imagine. These are probably the best applications most of the time that are sitting in the middle, right? But then who's tasked with evaluating it? That seems like a hard problem. I don't know if we have good solutions.
No, I think, of course, in all of these, we try to evaluate broadly, and we will send out things to other reviewers if we don't have the expertise. And I'm also not sure that interdisciplinary research projects are inherently better. There is certainly lots of field-specific work that is extremely high quality and very important. Maybe if I, can boil it down, I think that one rule of getting things funded is that at least one person on the panel needs to love your grant, and ideally 0 people on the panel hate it. And I think, so this thing about having someone on the panel love your grant can be a little bit more difficult if it sort of falls in between disciplines, right? If then say, well, this is interesting, I'm also interested in this thing. But if there's no sort of expert who can really see the value of these things, I think these things can be a little bit more difficult. This is obviously not a hard, fast rule observation, but I see that that's maybe where these kind of projects fail. I will not complain. We've been very fortunate at getting external funding for our projects, also the ones that bridge across disciplines. So, it's definitely possible, in particular, we're trying.
It can also be in the writing, right? This is something that's hard to quantify how good of a writer someone is.
Yeah,
it's hard to give an advice, just write well.
I give you grants on molecular biophysics where all the words and all the abbreviations make complete sense to me. And then there's another reviewer that's sitting saying, this is all nonsense to me. This person is not defining the terms, why they're writing this, right? So it's very much about the background of these things. That's unfortunately just the way it is. And you have to think about that, of course, when you write the proposals. But at the end, it is to some extent a lottery.
Right. And I think there is research that says that actually research that is more sitting across at least the disciplines ends up being more groundbreaking or transformative. But, well, as you say, it's not to complain about research that is within a well-established discipline.
And I also think that, I've seen some of these studies and it makes sense, but of course it still requires that we need some kind of metric for judging what good research is. And sometimes you can do this, right? But often these kind of large-scale analyses, I think they tend to boil down things to simple metrics of how were the papers, how many papers came out and how were they cited. And that is certainly a metric and it can be useful, but I think it's very difficult to address these things at a scale where you get statistics without relying on these kind of simplifying metrics. So I think the only real solution is to keep lots of diversity in funding and funding structures, ideally, of course, with as much funding as is needed to do the right kind of research, but then also just to make sure that we can fund big projects and small projects, interdisciplinary research, focused research for junior scientists, but also for mid-career scientists who, sometimes struggle enormously. I think, we just need this kind of diversity to keep, people, ability to do their jobs and use their creativity and good ideas for whatever it is that they're working on.
Absolutely. No easy solutions, but that's why it's worth thinking about. Okay, Kresten Lindorff-Larsen, thank you for coming on Space Invaders and sharing your expertise and your insights.
Thank you very much for having me, and thanks a lot for running this podcast. I'm enjoying it a lot, listening to it, so thank you very much.
Thanks so much. Have a great day. Thank you for listening. See you in the next episode of Space Invaders.